Hello everyone. In this lecture, I will be talking about screening of antibiotic producing organisms. But before we talk about the screening of antibiotic producing organism, I would like to tell you about the antibiotic. What is antibiotic? Antibiotic, it is the naturally occurring semi-synthetic or synthetic type of agent that destroys or inhibits the growth of microorganisms. This is the definition. Now I'll explain what do I mean by antibiotic. See, this let's consider this is one microbial cell and this is capable of producing antibiotic. Okay, so the microbial cell which is capable of producing antibiotic, and this is the another microbial cell. For example, we will consider it as a bacterial cell. Now this bacterial cell has undergone multiplication and therefore large number of bacteria are there. Okay, now the antibiotic which is produced by this living organism will act on the another living organism. Again, I will repeat, antibiotic produced by this living organism will act on these bacteria, and because of which this bacteria will die. Okay, this is known as the antibiotic. I mean, it is the substance which is produced by one living organism and it inhibits the growth of other living organism. That is known as antibiotic. Okay, I hope you have understood the definition of antibiotic. Now we will talk about primary screening of organisms which are capable of producing antibiotics. Now here, of course, we have a number of options to select as a source. We are going to consider soil as a source of microorganism for primary screening of organism capable of producing antibiotic. Now do the proper dilution of soil sample. Select the particular dilution of soil and spread it on the agar media. For spreading purpose, you take 0.1 ml of diluted soil sample. Incubate those plates at room temperature for 24 hours. Then after incubation, select the plates having colonies around 300 to 400. Now, after selection of plates, you observe for the isolated colony with zone of inhibition of other bacteria. Now, as we have selected soil as a source of microorganism, so soil contains different types of microorganism, isn't it? So here one in this plate, this is the picture where we can see different types of colonies. Now this is the one which is surrounded by zone of inhibition. Means if you carefully observe the in the vicinity of this colony, you will find the clear zone. Means something is produced by this organism and which has inhibited the growth of other organism. Okay, now select this colony and subculture it on another slant. And for further details like regarding the type of organism or details about this organism and details study regarding the antibiotic produced by this organism, for that purpose you send it for the secondary screening. You can see a few more colonies in this plates. Let's search if we get any other colony. Yes, you can see here also. You can see this is the very minute colony at the center, and the clear zone around that colony. One more colony is here. Very minute colony is at the center, surrounded by the inhibition zone. Means in the vicinity of this colony you will find the clear zone, that is the inhibition of growth of this other bacteria. Okay, but see, there is some disadvantage of crowded plate method. Whatever the method which just now we have discussed, that was the crowded plate method. See, this inhibition, whatever the inhibition which we have seen in previous slide, that may be because of production of acid or because of production of alkali, because this this picture or this criteria which we have used for selection does not give us clear cut idea regarding the cause of inhibition. That may be because of production of acid or alkali by the colony which is at the center, or it may be because of depletion of nutrient, that whatever the colony which is there at the center, that those cells might have used the nutrient at very high rate, and that may result into the depletion of nutrients. In addition to this, this method does not tell us regarding the inhibition spectrum. What do I mean by inhibition spectrum? That this whatever the antibiotic which is produced by the colony, whether this antibiotic is capable of acting on a large number of bacteria, or it is active against the small number of bacteria. Even this information, uh, I mean, this method does not give us the information regarding the inhibition spectrum. So for this kind of detail, we have to uh, do the secondary screening. But still, the at this stage, whatever the colony which is surrounded by the inhibition zone, you pick up that colony, subculture rate, means stick it on the nutrient agar slants or on nutrient agar plate, and use it for the further study. Thank you.